Hello everyone. Today we are gonna start the conversation talking about phospholipids, and this slide shows basically the structure of phospholipids and the different types of phospholipids that we have in cells and tissues. Basically, phospholipids, they are very important for formation of cell membranes, and we are going to discuss this a little bit more in detail in the next slide. But what I want to first show here is what makes up phospholipids. So on the left side here we have what we call phosphatidic acid. Phosphatidic acid is made up of, again, similar to what we saw with triglycerides. Here we have a glycerol backbone. Again, glycerol is shown here. So we have carbon 1, 2, and 3 of glycerol. And we have three fatty acids, two fatty acids that are bound or esterified to this glycerol molecule. So you have here on carbon 1, you have a saturated fatty acid. This is the way how phospholipids are actually made. So you have on carbon 1, you have a saturated fatty acid. It can be any, depending on the dietary source or what you eat. So composition of the diet will determine which type of saturated fatty acid you have here on carbon 1. Then on carbon 2, you have a non-saturated fatty acid that the same way as the carbon 1 position. In carbon 2, the type of unsaturated fatty acid will depend on the composition of your diet, so there are many of them. And then on carbon 3, if you were to look at a triglyceride molecule, you would expect another fatty acid to be bound here. But in phospholipids, it's different, because on carbon 3, you have what we call phosphate group. So this phosphate group really changes completely the way how the molecule actually now functions and interacts with your cells. So this phosphate group allows different molecules now to bind to it and form what we call phospholipid. So what we have here on the right side now is the 1, 2, 3, 4 different options of molecules that can bind to this phosphate group and form different phospholipids. So it would be, for instance, if choline, which is this molecule that we have here, binds to the phosphate group, we form phosphatidylcholine. If we have ethanolamine, which is the second molecule here, then it will form phosphatidylethanolamine. And if it's serine, which is this amino acid here, then it will form phosphatidylserine. And lastly, if we have this inositol group, which is rich in hydroxyl groups here, you're going to form the phosphatidylinositol molecule. So you have all these different forms of phospholipids in your cells, and it will depend obviously on which of these molecules is actually bound to the phosphate group. And if we only stick to this half of the molecule, that's what we call phosphatidic acid. When you bind this phosphatidic acid via the phosphate group, then you can create all these different 4 types of phospholipids that again are frequently found or mostly found as components of cell membranes. And why is that? Well, phospholipids, they are considered amphiphilic molecules, which means they can attract both water and fat-soluble substances. And this is a very important feature for cell membranes, because this membrane of a cell is a lipid bilayer. And because lipids and water-soluble molecules, they don't dissolve in each other. So you need to have something in the membrane that is made up of lipids that can actually talk to those or interact with molecules that are water-soluble, and also interact with molecules that are fat-soluble. So because phospholipids have this property, they can interact with both water and fat-soluble substances. They become very critical or crucial for the formation of cell membranes. So they are ideal structural components for cellular membranes. And also, they provide compounds. Example, arachidonic acid, which is an omega-6 fatty acid. for the synthesis of eicosanoids and for intracellular signaling in cell anchorage. For instance, phosphatidylinositol. So I'm going to go back to the previous slide just to show you. Remember, the carbon 2 position of the glycerol, you have an unsaturated fatty acid. In this case, if you have a diet that is rich in omega-6 fatty acids, specifically arachidonic acid, which is an unsaturated, polyunsaturated fatty acid, what happens is that if you release this unsaturated fatty acid, in this case is arachidonic acid, this can actually be used to form what we call mediators of inflammation. These are molecules that signal the cell to promote inflammation. And this is what we call, these products are called eicosanoids or leukotrienes. This is another group of inflammation mediators. So, and they come from the breakdown and the metabolism of arachidonic acid. So again, they going back to phospholipids in the membrane. And if you have a arachidonic acid sitting in this position here, so you're going to have more substrate to be used by the cell to produce these inflammatory mediators. And they are mostly located at the cell membrane. The other group of lipids that are really important too are sterols. And I think the classical one, and we are going to talk more about, is cholesterol. But sterols, they are a class of lipids that are characterized by what we call 4-ring core structure. And the name here is pretty long. I'm not going to ask you to memorize the name, but this is called cyclopentane perhydrophenantrine, or simply put, steroid nucleus. So the steroid nucleus is this diagram down here. So you have 1, 2, 3, 4. See, you have 4 rings that form the core structure that we call steroid nucleus. So from this basic steroid nucleus, we can form what is the most important of the sterol family molecule, which is cholesterol. So cholesterol is depicted here. So you see those 4 ring structures right here, or the sterol nucleus with a hydroxyl group here and a branching structure formed by carbons or hydrocarbon branching structure up here. So cholesterol, we can get it from 2 sources. It can come from the diet, for instance, when you eat any animal product, because cholesterol is only found in animal foods. It's not found in plants. Plants have a similar but different type of, it's called ergosterol. It's not what we see here. Cholesterol is essentially an animal sterol. And you find it from the diet when you eat meats in general, when you eat egg yolk. It's famous. Eggs are famous for being rich in cholesterol. Poultry, and also in dairy products. So these are mainly the sources that you get dietary cholesterol. But in case you decide not to eat any cholesterol, you can say, oh, I'm going to ban completely any source of animal foods. So I'm going to be a vegan or a vegetarian. I'm not going to eat any animal product. So then you don't get any cholesterol from the diet because you're not going to get it from any plant. So what happens is that your body makes it. So cholesterol is such an important molecule for your system, for your organism, that you have to have it present all the time. So your body is prepared to make it in high amounts in case you don't eat it. Now, if you eat, your body makes less. But if you don't eat, it will make more. So this is what we call endogenous synthesis, and we are going to discuss it in a moment what this pathway of endogenous cholesterol synthesis is about. But here it is. So why is it that we say that cholesterol is such an important molecule? Because using cholesterol, you can, or your body, by using cholesterol, cells in your body, by using cholesterol, you can make bile salts, or bile itself is essentially made up of cholesterol. So right away, you see the importance of cholesterol for digestion of lipids in your body. Forms steroid hormones, testosterone, estrogen. These are all derived from cholesterol. Cell membranes. You have in the cell membrane molecules of cholesterol that are actually localized within specific sites, specific niches within the cell membrane. You also have lipoproteins, and we are going to discuss them in detail, but lipoproteins are the molecules that actually transport lipids in your blood. So you have there also cholesterol. And lastly, but not the least, vitamin D3. So vitamin D, which is an essential lipid-soluble vitamin, it comes from cholesterol. Remember, if you don't eat vitamin D from your diet, you can actually make vitamin D. And how do your cells actually make vitamin D? By using cholesterol, transforming cholesterol into vitamin D3. And again, we are going to learn about this in detail when we get to vitamins or lipid-soluble vitamins. But this gives you an idea of how important cholesterol is for normal function of the organism. And it is by far the most common steroid in animals. And just to give you also an idea of how crucial it is for you to have abundant cholesterol in your body, is because in the brain it's extremely enriched, or the brain is extremely enriched in cholesterol. Also the nerve tissues. So you can't form proper neural tissue, or your brain cannot be not only function, it cannot be formed properly, and then therefore cannot function properly if you don't have lots of cholesterol in it. So again, just to synthesize here, or to make sure that even though cholesterol is frequently vilified because we associate cholesterol with cardiovascular disease, it's not a molecule that we can actually eliminate. We shouldn't, and we need it in significant amounts in our body. And again, if you don't eat, your cells will make it. So how does that happen? So how do cells make cholesterol? So there's an extensive pathway or biochemical pathway that does that. So it's kind of synthesized or abbreviated here, so just for easy of us to understand. But for instance, you can take fatty acids, and you can take glucose, or even amino acids, all these three molecules that are abundantly present in the foods that we eat, and you can convert them into acetyl-CoA. And I'm sure that you all have heard about this molecule, acetyl-CoA. It's commonly associated to the Krebs cycle, because that's where acetyl-CoA is usually complexed with another molecule called oxaloacetate and forms citrate, and then it cycles within the Krebs cycle, and that's where usually acetyl-CoA is mentioned in our body. But in fact, acetyl-CoA doesn't have necessarily to be used in the Krebs cycle. Acetyl-CoA can be used for many different reasons. One of them is that your cells take acetyl-CoA, converts through a series of reactions that you don't have to memorize. You don't need to worry about this, but you need to know that acetyl-CoA is converted into a molecule called HMG-CoA, or hydroxymethylglutaryl coenzyme A. So this is a crucial molecule because it's an intermediary step, or it's a rate-limiting step here, where HMG-CoA is converted into mevalonate. So the conversion of HMG-CoA into mevalonate is what we call rate-limiting step in the process of cholesterol synthesis. And obviously, once it passes through, it becomes mevalonate. There is another set of reactions, becomes squalene, and then squalene through another set of reactions become cholesterol. So cholesterol can be ultimately formed from glucose, fatty acids, and amino acids. And it goes all the way through this pathway to become cholesterol. So why I want to just spend a couple minutes here just talking about this rate limiting step, because it's called HMG-CoA reductase, the enzyme that actually catalyzes the conversion of HMG-CoA into mevalonate. So in other words, for you to make this rate limiting step, you need this enzyme. In normal cells, what happens is that if you don't eat cholesterol, your body needs it, so it's going to say, well, let's make cholesterol. So this whole process starts and goes all the way to cholesterol production. So what happens is that once cholesterol inside of the cell reaches a certain amount, a certain concentration, the cells detect it and says, well, we need to stop production of cholesterol. So you need to be able to switch on and off depending on how much cholesterol you need. You don't want to just be producing cholesterol endlessly. You need to have the capacity to switch on and off the process. So how do the cells do it? So normal cells, usually as cholesterol is being produced and it accumulates in the cell, it has a negative feedback mechanism that shuts down the enzyme called HMG-CoA reductase. And again, by shutting down this enzyme, it stops the conversion of HMG-CoA into mevalonate. For that reason, now it shuts down the whole process of endogenously production of cholesterol. So that's the way how normal cells usually operate, to make sure that you have enough cholesterol being made available in your body for any function, as we mentioned before. So normally, under normal circumstances, in normal people, liver makes approximately 800 to 1,500 milligrams of cholesterol every day. And this is made through or via this pathway that I'm just showing here. So another important thing about this endogenous cholesterol pathway synthesis is that knowing that this enzyme can be regulated now, what the pharmaceutical industry did is that they created a drug that you can actually voluntarily stop the production of cholesterol by inhibiting this enzyme. And that's what these statins actually do. It's one of the most popular drugs, most prescribed drugs that we have nowadays, especially to lower cholesterol levels in your body, because HMG-CoA reductase can be inhibited by a drug. So when you take, or if anybody in your family is taking a statin, a famous one, for instance, is Lipitor, this one will actually inhibit, is an inhibitor of HMG-CoA reductase. So again, in normal cells, this process works well, but it looks like in certain people, this ability of HMG-CoA reductase to be inhibited by cholesterol is for some reason lost. And what happens is that people keep producing lots of cholesterol. They don't stop, they don't put a brake, or they don't switch off the production of this sterol. For that reason, what the pharmaceutical industry does is, well, let's say if your body is not doing it by itself, let's do it pharmacologically. So you take a drug that goes there and suppresses the activity of the enzyme. So this pathway is existent. Every cell in your body, almost every cell in your body has the capacity. Liver does quite a bit, quite very well. It's a very potent producer of cholesterol, and you know why. One of the reasons is because it needs to synthesize bile on a regular basis, and bile is made up essentially of cholesterol. So this pathway is very active in the liver. And what is important for you to know is that it can come from glucose, fatty acids, and amino acids, producing acetyl-CoA, and you have a rate-limiting step that is the conversion of HMG-CoA into mevalonate, and the enzyme HMG-CoA reductase is crucial to regulate this process. Now, also on the topic of cholesterol, there is another thing that I want to discuss with you that is called enterohepatic circulation. So what is it? It's the process by which bile salts are reabsorbed in the gut. Remember, if you release bile salts in the duodenum, it will do whatever it's supposed to do. In this case, it works as an emulsifier. It breaks apart those large droplets of fat. So now bile acids, once they do what they're supposed to do, they're not completely eliminated. Actually, most of the bile salts are reabsorbed into the gut, because if you lose all the bile that was, all the bile salts that were released in your gut, it would take a lot of work for your liver to keep producing more bile all the time, because you would be losing it. But in order to prevent that and make sure that you always have bile available in your system, there is larger reabsorption of the bile that is released in the gut. So here it is, the diagram that kind of depicts what's going on here. So we have cholesterol in the liver, so liver here in pink. So liver makes bile. So bile is rich in, again, cholesterol, and you have cholesterol in its free form, which is not a lot, but it's to some extent you have some free cholesterol that has not been modified at all. But the vast majority of the bile was actually converted into bile acids and bile salts. So now once this product, this solution is released in the duodenum, and specifically in your intestine, obviously, it will do what it's supposed to do. And then the free cholesterol, approximately 50%, which is not a large amount to begin with, but 50% of whatever was there goes out in your feces. So there is some excretion, some loss of free cholesterol in feces. And the other 50% goes back, is reabsorbed, and goes back to the liver. Now, bile salts, 97% is reabsorbed, and you only lose 3%. So the vast majority of bile salts go back to the liver through what we call enterohepatic circulation. This is the mechanism by which your body finds or uses to actually sustain or maintain most of the cholesterol or the bile salts that produces and that is released on a meal-to-meal basis. So again, even though your liver produces quite a bit or uses quite a bit of cholesterol to make bile, there is a large amount of that bile that is reabsorbed, because that kind of takes a little bit of the pressure from the liver of producing cholesterol all the time. So you need to be able, so it minimizes, let's put it this way, the rate of production of bile if you were to eliminate everything, all the bile, every time that it's released in the gut. So it doesn't happen, so it facilitates, so your liver constantly has a certain supply or a storage of bile in it, or at least not in the liver specifically, because the liver transfers it to the gallbladder and then the gallbladder releases it. But it's important that this is reabsorbed, goes back to the liver, so the liver can manage this more easily, or the production of bile more easily. So this is called enterohepatic circulation. Now, let's try to establish a relationship here, because I've been talking about cholesterol and showing all the importance of having cholesterol in your body. Now, but there is again, cholesterol has been vilified for a long time now, and in most cases people want to actually eliminate or reduce cholesterol from the diet as much as possible. So where is this coming from? So how this whole thing started? So this starts with what is called the diet heart hypothesis. Early in the 1960s, that's when 50s and 60s, when this diet heart hypothesis actually came up. And the idea was this: if you eat saturated fat, and this is depicted here by meat, poultry, and dairy products, these are animal products that contain saturated fat. And what happens is that when you eat saturated fats, your cholesterol levels or cholesterolemia go up. And then the direct association was that this would cause cardiovascular disease. And that is the basic premise of linking, or that linked fat with cholesterol, or saturated fat with cholesterol, and cholesterol with cardiovascular disease. And to actually put forward this idea, there were studies where people or scientists actually looked at the composition or what happens with atherosclerosis in an artery. So what we have here on this right side here, this is a diagram that shows different degrees of atherosclerosis. In other words, accumulation of plaque in an artery. So what we have here is a demonstration of three different things. So on the left side here, what we have is in purple and kind of whitish or grayish color. So what we have here is a section of an artery, and this is divided. If you were to look at the artery, if you do a cross section and you put it on a slide, this is what you would see. So this upper portion that is in purple, this is what we call inner membrane, or abbreviated here as I. That's what I here in this figure indicates. And M is medium. So why is I is intima? Because intima is the layer of cells on your artery that is in contact with your blood. And we say that it's the innermost layer of cells that makes up the structure of your artery. So if you were to look on top here, that's where blood would be circulating. If you were to look at a circular structure, three-dimensional circular structure, which is your artery. So blood is flowing up here. So the contact of blood is with the intima layer. And you see here, when there is no lipid deposits, when it's a healthy, free of disease situation, you see a very thick intima layer, purple. and this is the medium, is the layer below it, where you have smooth muscle cells that really allow the artery to contract and dilate. So this is a smooth muscle. And there is a third layer here that is not shown here very well, but it's the adventitia, which is the layer that is more fibrotic or gives strength to the artery, and actually envelopes or encapsulates the other two layers. So the adventitia would be the outermost, media would be the one in the medium, in the middle, exactly in between the adventitia and the intima, and then you have the innermost layer, which is called intima. So now that we know this, we have on the right side here, or right to the right of this first column, we have the amount of lipids that you can see there. And then also the infiltration of macrophages. Macrophages are cells from the immune system that infiltrate that when there is inflammation, an inflammatory response, for instance. So what happens is that in a normal, healthy, free of disease situation, you see that the inner layer or the intima is very thick. There is basically no lipid infiltrated, so it's clear. You see that there is no signs of lipids here, and the arrows here are indicating actually the transition between the intima and the medial layer. So here in the staining, you can see that either in the intima or the media, you don't see any presence, any signs of lipids, and you don't see infiltration of macrophages either. But as the disease or the accumulation of plaque progresses, we have what is called grade 1, when some lipids start to show up here. So what happens is that the colors start to change a little bit. There you see a little bit more of the whitish, grayish color present in the purple. And then when you look at it and you stain for lipids, you see that it's starting to get red. This is red staining that really indicates the presence of lipids. And at this stage, you have an infiltration of lipids, as you can see here, close to the media, at the transition of media and intima down here, starting to accumulate lipids. And still, you don't see any macrophages because doesn't seem to be a very strong inflammatory response there yet. And then, if you look at grade 2, we have more lipids again. The red becomes more evident. It's still very little of almost no macrophages in large amounts infiltrated here. But then when you go to grade 2 in a later stage, you see that there is quite a bit more lipids. See the incidence or the dark red here, the amount of red that you have. And now you start seeing also macrophages that are infiltrating because there is a typical inflammatory response that is taking place there. And then we have here grade 3. You have much more spread amount of lipid within the entire layer of the intima layer, and it's starting to get actually into the media too. You see that there is infiltration crossing the line between the intima and media, and these are lipids, and then again a lot more macrophages. And grade 3, when it's really pathological, when there is thickening with foam cells, you see here that it's white material. And this is really a large accumulation of lipids here and a large accumulation, also infiltration of macrophages. So this is by looking at this and understanding what's going on here, at least identify the composition or elements that make up this plaque. That's where fat was associated with cardiovascular disease, more so cholesterol, because if you take a sample of these layers here and you analyze the type of fat, you're going to see that it's a combination of macrophages with fibrotic tissue and also cholesterol. For that reason, there is this association of cholesterol with heart disease. It doesn't mean that cholesterol was the problem that began the process, but because cholesterol is present, then cholesterol was associated as the initiator or the molecule or the element that actually started the process, which is nowadays we know that it's not quite the case. There are other issues associated with the development or accumulation that leads to accumulation of lipids here that are not necessarily dependent only on the amount of lipids present in the blood. There are other things that lead to that, but that's a discussion for another day, another course, not for here now. For now, what I want to show you is the relationship between what we say the diet heart hypothesis and how it was kind of reinforced by the fact that lipids are present in these layers of the artery. So now if we look here, and then again to shed a little bit more light and link this to nutrition also, is that if this graph here shows us on the left side we have the consumption in grams per capita per day. In other words, how much of fat, in particular case saturated fat and vegetable oils, took place from early on in 1900, so the beginning of the 20th century, all the way up to now, pretty much close to what the current time that we are living. So, and then this is time in decades in the x-axis, and on the right side here we have heart disease in deaths in thousands of people. So if you look here, let's look first at saturated fat. If you look at the consumption of saturated fat, this is in triangles. This, if you look at the curve that saturated fat from the beginning of the early on in the 20th century, if you go all the way down to current days, or very close to what we are now, to 2010, you see that there was very little alteration in the intake of saturated fat from the diet. So people didn't increase drastically saturated fat. That you say, well, there is a relationship. This is the culprit or the responsible for the development of cardiovascular disease. Now look here, the dashed line shows us what the heart disease deaths is. So if you look here, go back, you see that up to 1910, 1920, it was relatively low. It was only about 100,000 people that were dying at that time. And then when you look at this, after that time, there was a progressive increase. And this is where we are nowadays. We have what, 600, 700, between 600 and 800,000 deaths related to cardiovascular disease or to heart disease. Now, it doesn't correlate at all with saturated fat intake. If anything, it correlates more with vegetable oils. See here, it follows almost a very, very similar pattern. Early on, up to 1910, 1920, we didn't see much, but then it was progressively increasing. And then after 1960, remember when we discussed previously that the American Heart Association and also the guidelines for dietary guidelines in the US, they actually really promoted the use of vegetable oils. And most of it is actually seed oils. Look at the sharp increase. And that's actually what follows more likely towards or similar to the pattern of heart disease deaths. So if anything, it's not saturated fat that seems to correlate with heart disease. It's more so the intake of vegetable oils. And especially, remember when we spoke about soy oil that is rich in omega-6 fatty acids and more inflammatory and with all those associations with cardiovascular disease. So this is just to illustrate and give you an example that not everything that we hear or everything that we think seems to be relevant for cardiovascular disease, especially when we vilify fat, saturated fat, and we vilify also cholesterol. It doesn't seem to be so, or it doesn't hold water so much, because there seems to be a lot more to it that we don't actually understand well. But it was, and it's popularized the idea that it's saturated fat, but it doesn't seem to be, the relationship is not, doesn't seem to be there. Now, why is it that this vegetable oils or seed oils seem to be more problematic? Actually, it's these oils, they are rich in monounsaturated and polyunsaturated fat. And this graph here shows how the oils turn toxic. Toxic is because it produces substances that can be really nasty and toxic to your cells. And what they did here is they took different oils, coconut oil, butter, extra virgin oil or olive oil, and corn oil and sunflower oil. So these are, with the exception of butter, these are all vegetable oils. And obviously coconut is very saturated, and then you also have butter, which is also rich in saturated fat. And you see here that these are the ones that normally are solid at room temperature. And then you have the extra virgin olive oil, corn, and sunflower. So what we have here on the left is the release of toxic aldehydes by heating up these different oils. So what happens is that if you do 10 minutes, and this is at 180 degrees, so you're going to put oils, and all these oils were put on a frying pan, and then you heat up at 180 degrees, and then you measure how much of these toxic aldehydes comes out of these heated oils. And the more mono and polyunsaturated fatty acids you have, more double bonds, and these double bonds make them to be more susceptible or more vulnerable to oxidation. That's the idea. So look here, if we compare coconut, butter, olive oil, corn, and sunflower, when you heat approximately 10 minutes at 180 degrees, you get a very small amount from the saturated fat oils. And you get a significant, much higher, it's still not too much, but much higher release of this toxic aldehydes than you get with saturated fat. If you heat it up for 20 minutes, look at the drastic increase. So then you have coconut oil, the most saturated fat that you have, with an increase, but it's still relatively small. Then there is butter, and then all the vegetable oils going up at a much higher rate than you get for the oils or the fat that it was saturated. And then if you heat up for 30 minutes, then you get a massive increase, especially in this vegetable oils or the seed oils, as opposed to almost no change in the release of these toxic aldehydes that you get from the heating of more saturated fat. So what's the message of this slide? The message here is this: if you're going to cook, if you're going to fry something, avoid using vegetable oils. Try to give preference to those fats that are more saturated, because they are more stable under resting or room temperature, and also under heating conditions. And the more you heat and the longer you heat, the worse it gets. And some people have associated this production of toxic aldehydes to the development of cardiovascular disease, inflammation, cancers, and all different types of diseases associated with this toxicity that arises from the release of aldehydes once you heat this mono and polyunsaturated fatty acids that have this lots of double bonds, or either one or multiple double bonds, and they are more susceptible to oxidation and then the production of these toxic aldehydes. So that's the message. So when you pick the oil you're going to cook on a regular basis, if you're gonna heat up, try to use oils or fats that have more saturated fat. Now, if you're gonna eat a salad, then it's fine if you use extra virgin olive oil, which is what most people usually use. Avoid always all other seed oils. You stick to extra virgin oil. I think it's a good idea, because it can give the taste, the pleasant taste you want in your salad. You're not going to heat it up because your salad is eaten at room temperature, so you won't have much of an issue with the production of any of this toxic aldehydes, because you're going to consume it at room temperature. So for a salad is one thing, but if you're going to heat up, if you're going to cook, then keep in mind that you don't want to really use those unsaturated fats that can really produce toxic aldehydes. So from this point on, I want to really switch gears, and I want to talk about the digestion and absorption of lipids. Because up to now, we are talking about lipids is in their general form, what they are, how they are made up, their importance. But we have to keep in mind that when you eat fat, you have to digest to be able to absorb. So how is the digestion and absorption of lipids organized? So lipids, as I said, are generally not soluble in water, and the enzymes that digest them are secreted in the aqueous solution. In other words, in your duodenum, in a solution that is basically made up of water. So a specialized system needs to be in place to allow lipids to be accessed by their respective digestive enzymes, and also for the absorption and transport into the circulation. This is what we spoke also early on in this course, the need of existence of a specialized system that will actually allow lipids to be processed in our body in an aqueous solution. So the process starts when you're chewing in your mouth, because you release what's called lingual lipase. Lingual lipase starts breaking down triglycerides, especially separating those fatty acids from glycerol. So the breakdown is occurring, begins there. It's relatively small amount, because the time and the time that food stays in your mouth is relatively short. So there is a beginning of the process, but it's not the massive. It's a limited amount of lipase activity that you get there. So then the process continues in the stomach, because you also have gastric lipase. You swallow the food, food gets into the stomach. Stomach, not only hydrochloric acid, but you also have peptidases and gastric lipase. So that also accelerates or continues that process of breakdown of lipids. And mostly we are talking about here is triglycerides, which is the most abundant kind of fat that you have in your diet. And remember, triglycerides are 3 fatty acids with one backbone of glycerol. You need to separate those, because you can't absorb that huge molecule. You need to separate fatty acids from glycerol, and then you can absorb them separately. And then later on, you can put them together again and make another triglyceride molecule. But you need to be able to separate them first, make them into smaller molecules so they can be absorbed by the gut. So lingual and gastric lipase, they begin hydrolyzing preferably triglycerides containing short and medium chain fatty acids. And this depends on your diet. Depending on what kind of food you're eating, you may have different types of fatty acids making up that triglyceride molecule. It could be short chain, could be long chain, could be medium chain, so different ones. But this lipase in the mouth and in the stomach, they preferably cleave short and medium chain fatty acids that are present in triglycerides. So fat entering the duodenum, once it passes the stomach, it's going to go into the duodenum. So it's made up, that fat that is in that chyme is made up of approximately 70% triglycerides, with the remainder composed of a mixture of partially hydrolyzed lipid products. So fatty acids that you have that were cleaved, and a large amount of DAG, which stands for diacylglycerol. It's fatty acids in position 1 and 2 of the glycerol, or carbons. Fatty acids are esterified to position 1 and 2, or carbon 1 and 2 of a glycerol molecule. So that's what we call DAG. So this is basically what enters, or the majority of the fat that enters the duodenum, because the other components were derived from the cleavage of these triglycerides that happened to some extent in the mouth and to some extent in the stomach. So now remember that I mentioned lipases and co-lipase. So along with pancreatic juice, so in the pancreatic juice you have lipase now, because it's what releases into the duodenum to continue to actually finalize into the bulk of the digestion of lipids. So you have their lipase and you have co-lipase. And also you have the release of bile salts, bile acids from the gallbladder. So everything, chyme is arriving at the duodenum, and all these juices are being released on top of it. So it turns out the bile salts, in the beginning, they inhibited the lipase activity because they displaced the enzyme from the surface of the lipid droplet. Remember, because bile emulsifies, breaks a lipid droplet, this big globule of fat, into millions of thousands of those tiny lipid droplets. When it does that, it ends up displacing the lipase that was there, the lipase that was actually trying to break down the lipids that were there, because this lipase came from the mouth or came from the stomach, so they should be there. But the problem is that now it's completely displaced, plus there is lipase that comes in from also the pancreatic juice. So in order to try to solve this, or to circumvent this problem, pancreatic juice also contains co-lipase. So it binds lipase and reverses the inhibition by bile salts. So bile salts took them apart. Now co-lipase binds to lipase and brings them together again to the lipid, to the surface of the lipid droplet, and allows lipase then to continue cleaving the triglyceride molecule. So again, there is a synergistic or a cooperation between lipase and co-lipase, because lipase does the actual cleavage, but you also need co-lipase to maintain lipase attached or attacking the lipid surface of the lipid droplets, even after you have this displacement of this lipid droplet by emulsifier, in this case biosalts. So this way co-lipase ensures adhesion of lipase to the lipid droplet, so lipolysis can effectively occur. So now this is a diagram that tries to put together the digestion that is occurring in the duodenum, especially when you look at the intestinal lumen. So here in pink, what we have is an enterocyte. This cell you already know because we discussed this, we spoke about this, you saw this when we were doing digestion of carbohydrates. So again, we have here the brush border that is made up by the microvilli. So this is depicted here on the top of the cell, or the apical portion of the enterocyte. And you can see here that we have the intestinal lumen. So this is the place where all the chyme is being digested. So bile acids are released. And along with it, you have lysophosphatidic acid, and you also have some free cholesterol. So what happens is that the presence of bile acids, this is the lipid droplet that contains triglycerides, remember, and some of the triglycerides that have been digested, 1,2-DAG or diacylglycerol. So this is one of the droplets. That what happens is that when bile was released, it emulsified this big lipid droplet and separated them into smaller molecules. So with this, pancreatic lipase now comes in and really breaks apart or starts chewing off and separating these different components that make up the big lipid droplet. So emulsification was important because instead of having one big globule, now you have tiny little ones, and then it's easier, you have a higher surface area, easier for access for the pancreatic lipase. So it chews this down or breaks this in smaller pieces. So you're going to have what we call long chain fatty acids, LCFAs. These are fatty acids alone on their own because you chop them off of the glycerol or the glycerol backbone, so they're separate. Not all of them are. You still have some that remain attached to the glycerol backbone, and this is the position 2, so that's why it's called 2-MAG. MAG stands for monoacylglycerol. So 2 because it's the position 2 of the glycerol molecule, that you still have one fatty acid attached. You also have 1-MAGs, that is, you took off the position 2, but the fatty acid present on the, or esterified to the carbon 1 of the glycerol still remains. So it's 1-MAG because it's the carbon 1, 2-MAGs is because it's carbon 2. And also you have some long chain fatty acids that are completely free from glycerol. So all this together. So now you also have some cholesterol, free cholesterol, that is present in the food, because the food is, if you ate a nag, for instance, you have cholesterol there. If you eat chicken, you also have some cholesterol. If you eat meat, you have cholesterol there. So the food is being digested, so you're separating all these components. So now they are all together. And again, because you have bile acids and salts present in this mixture, they really keep also all these molecules that are fat soluble, because they tend again to stick together and form another big globule of fat. So again, because bile acids and salts are present there, they break them apart again, or they keep them separate. And that's what we call micelles. And these micelles are really tiny droplets of this lipids, this composition with lysophosphatidic acid, cholesterol, 1-MAG, 2-MAGs, and long chain fatty acids. This is all together now forming tiny little droplets called micelles. And these micelles, they can actually cross the membrane of the enterocyte That's where the microvilli or the brush border, they enter. So now what you have is inside of the enterocyte, remember you have that layer of enterocytes that line up the surface of the duodenum. So now all these molecules, tiny molecules now are going in. So you have again 1 megs, 2 megs, long chain fatty acids, and cholesterol. Now, what your enterocyte does here is okay, we broke apart all these big, large molecules, so the triglycerides, we broke them all apart to be able to form these smaller molecules that can actually cross and enter the enterocyte. So now, inside of the enterocytes, what happens is that they are put together in a structure called chylomicrons, where you combine, again, 1 meg, 2 megs, and long chain fatty acids, and you make them again back into triglycerides or triacylglycerol molecules. And then you add cholesterol, and you also add lysophosphatidic acid or phospholipids, anything that is present there, and you form this big, or this molecules, or these particles, not molecules. Let me make sure this is a particle that contains all these different fats assembled, and we call them chylomicrons. So this is what arises inside of your enterocyte. And also, there is one protein called ApoB48. ApoB48 is a protein that is synthesized within the enterocyte, and for every particle of chylomicron, you have 1 ApoB48 on top. It's like as if you had like a circular structure, which is the chylomicron, and on top you put a flag, and this flag that is hanging on top of this particle is ApoB48. It stands for apolipoprotein B48, and 48 because it's the molecular weight. 48 kilodaltons is the size of this protein. So every chylomicron has 1 ApoB48, so it's a 1-to-1 ratio. So now you assembled this whole particle called chylomicron, rich, very rich in triglycerides, because it's the most abundant type of lipids that you have in your food. And obviously you also have a certain amount of cholesterol, a little bit of phospholipids, phosphatidic, lysophosphatidic acid, or any other lipids, but the bulk, the majority, by far, you'll see later on, that is made up of triglycerides. So now remember that I mentioned the lymphatic system. So the green tube that is depicted here is what we call lacteal. Lacteal is that branch or that portion of the lymphatic system that actually irrigates the villi, that allows the exchange between enterocytes and your internal environment. So that's where these particles actually go first. So when we spoke about carbohydrates, we mentioned that glucose, for instance, lactose and fructose, they all get into the circulation through the regular blood vessel. Now, chylomicrons, they enter via lymphatic system, and this is a lacteal branch where they get in. So they cross the enterocyte, the basolateral membrane of the chylomicrons, of the enterocytes, and then they enter the lymphatic system, and then they eventually get into the circulation. Remember, the lymphatic system drains your gut, and then it goes all the way up to your thoracic duct, and that's where it communicates with the blood, and then there goes fat inside, or these chylomicrons are delivered to your body, so it can actually get to the cells, and the cells can actually use all these lipids that were originated from the diet that you just, or the meal that you just had. So the absorption of lipids in the intestine depends on these micelles that interact with the microvilli at the brush border, and then allow the movement of lipids into the enterocytes via protein-dependent and independent processes. So we have here the duodenum and jejunum. So these are the sites where most of the lipids, especially the duodenum, is absorbed. So it can go in it can cross the membrane and enter the enterocyte by diffusion. In other words, there is a difference in the gradient. There is more in one area than the other, so it goes from the higher concentration to the lower concentration. So it's from the lumen towards the interior of the enterocyte. But you also have active transporter mediated by transporter. So you have proteins that actually do that job. So you have FATP1 and 4, or ATP, a fatty acid transport protein. You have 1, 2, 3, and 4, so you have 4 different types. And there is another one called CD36, which is cluster of differentiation. That's what CD stands for. But we now know that this is also CD36 because there are several of these clusters of differentiation there. So, but this one specifically works as a transporter of lipids. So together they allow all the lipid to enter the enterocytes. And for cholesterol, there is one called NPC1L1, which is Niemann-Pick cholesterol transporter here specifically. Niemann-Pick is the name of the person that actually discovered this. So this is one transporter that is specialized in cholesterol. So it allows cholesterol to cross the membrane and get easily inside of the enterocyte. So 2 different mechanisms. One is by diffusion, but you also have transport mediated, because both of them maximize the ability of the enterocyte to take up fat. So now once they get into the circulation, we have the transport of lipids in the blood. So we have again chylomicrons because they come from the intestine. We also have in the blood VLDL, which stands for very low density lipoprotein. VLDLs are produced in the liver. We have LDL, low density lipoprotein, that what happens is that once VLDL donates its fat content to the cells, it becomes LDL. And we have another one that comes from liver that is called high density lipoprotein, or HDL. So this is also from liver. So we have 2 particles that arise in the liver and one that arises in the intestine. Obviously, from a dietary point of view, from a nutrition point of view, it's the chylomicrons that transport all the fat that we get from the diet, because this is where the fat is digested. It's in the intestine that it is organized in a particle, and that's where this particle is formed and transferred into the circulation to be used by cells and tissues. So chylomicrons is crucial for dietary fat. VLDL is the fat that is produced in the liver. So that's the way how this arises, or where it comes from. LDL is a product of VLDL, so it's a conversion of VLDL into LDL. And HDL is an entire separate set of particles that have their specific features but also come from liver. And this is the composition of these different types of fat, of lipoproteins. So again, chylomicron is the biggest one, and it's basically in yellow is the composition of triglyceride, the content. See, it's almost 90% triglyceride, very little cholesterol, approximately 5%, I would say. Phospholipids is even less than that, and protein is very little, both basically triglyceride. VLDL is the second bigger, or second in size. So again, reaching in triglycerides, but it comes from liver, not from the gut. So it has a little bit more cholesterol, as you can see here. It's approximately 10%, about double the amount of cholesterol that you have in chylomicrons. Same thing with phospholipids, and protein a little bit more. And then you have LDL that has very little triglycerides now. It's the opposite. So now you have approximately 10% here, I would say, triglycerides. The bulk of it is approximately 40% is cholesterol. Then we have, phospholipids and protein. And the smallest particle of all of them, which is HDL, that has almost no triglyceride, has approximately 20% is cholesterol, rich in phospholipids, and even richer in protein. So again, in size, they vary from the biggest one, which is chylomicron, to the smallest one, that is HDL. And obviously the difference is that as you reduce in size, you lose triglycerides, and you gain proportionally in the other components that make up the particle. And you don't need to memorize the diameter, density, because I'm not going to ask you questions specific about numbers. So don't worry about that. What you need to know is the relative composition of them all, knowing the difference between a chylomicron, for instance, and an HDL and an LDL in terms of what kind of composition is more prevalent in one versus another. So you need to know that relationship, but not these specific numbers. And I'm not going to ask you to memorize the diameter, the density of each of these particles. But obviously you need to know, it's obvious that the more fat you have, the less dense it is. The more protein you have, the higher the density. And so that's exactly what it says down here. Okay. And we also have, to finalize this, is the role of apoproteins. So every lipoprotein has apoproteins. So there is not only that fat core, but you have on the surface of that fat structure or fatty particle, you have also proteins sitting on top of it. And that's what we call apoproteins. So why are they important? So remember I mentioned ApoB48. ApoB48 was one flag on top of a chylomicron. So what is that? What are these apoproteins important for? So they're important for the distribution and exchange of lipids in the body, and also for the well-controlled processes that orchestrate how the exchanges occur. So the apoproteins, or apo, these are the ones that dictate the fate of lipids in your body. In other words, as the particles travel in your system, the apoproteins are the ones that actually signal where they should go, which cells will take them, which cells will collect the material that they carry, and also it will tell the particles to go to different places, to different directions in your body, specifically not only for donation of lipids to tissues, but also for the removal of these particles from the circulation. And we are going to learn this as we go, as we progress here. So apoproteins, they confer water solubility, and they regulate the activity of key enzymes in lipoprotein metabolism. We are going to study this more in detail. They mediate particle removal from the circulation by binding to specific receptors located on the cell surface in various organs and tissues. Remember, you have a particle circulating. How is it that the cells know what's going on? It's because you have this proteins sitting on top of these particles that actually bind to receptors or communicate with the cells. And then once that communication is established, it allows the exchange between the cells and the tissues, and vice versa, the tissues and the particles. The particle with cells and tissues, and also between the tissues and the particle, because every time that the particle anchors or interacts with a cell, it will not only transfer stuff, but it may also collect things from the tissue. So that exchange is regulated by all these apoproteins. So we have 4 lipoproteins we are going to discuss. So chylomicrons, they contain ApoB48, and they originate from the intestine. So ApoB48 is the apoprotein that is sitting on top of chylomicrons. VLDL and LDL, they have ApoB100, and they come from the liver. ApoB, there is only one ApoB per particle, and it functions as a receptor ligand, as we mentioned before. There is also ApoE, and there are 3 different isoforms, and is present in almost all lipoproteins. And HDL contains apoproteins A1, A2, A4, and C. So there are several different apoproteins. You don't need to memorize them all. We are going to just discuss a few of important ones, so you know how the fat that came from the food that you ate, how it ultimately gets to the cells, and how this process occurs. Because you need to have this ability to communicate with cells, and as these particles travel through the blood, they need to be able to interact and communicate with the cells. And these apoproteins, that's what they do. So with that, we are going to stop today. We are going to next discuss each of the lipoproteins. We're going to start with chylomicrons, and then understand exactly how they are processed by the cells and tissues in our body. And also not only the nutritional implications, but also there's some potential pathological implications associated with these lipids. But this is for the next lecture. Thanks for watching, and bye now.